In practice, we work backwards a bit. We start with the tissue and we say, what's happening here as you progress from health through disease? We use an approach that we kind of say is like the Framingham Heart Study, right? That was an incredible longitudinal study that showed how do we monitor health through different stages of disease. Now, took forever, and in today's dollars, I don't know, billions and billions and billions of dollars, but it manifested in one of the biggest drugs or biggest impacts in the world, in statins and identification of cholesterol. And we thought, with these technologies that we have today, is it possible to do that instantaneously? And so, with effective subclinical labels, with access to population scale tissue and blood, we've been able to figure out at the single cell level, at the tissue level, what are the cascading biological events that are leading from health through different stages of disease, ultimately through end stage disease? How do we figure out which of those are going to be presenting themselves as potential biomarkers? Really, circulating proteins is what we're focusing on. And then which of them can we use our causal inference models to identify are therapeutically relevant? And maybe a little bit, if we're talking a little more futuristic, how do we think about combinations of them that can actually bring people from any specific stage back into fully functioning, regenerated organ or person?